nuclear DNA, biparental inheritance, comes in a chromosomal structure, so you have all that secondary and tertiary structure being packed in. With mitochondrial DNA, it's a little bit different. This is our ancestral relative back from the bacterial days. Mitochondrial DNA is only inherited through your mom, maternal inheritance. So I have the same mitochondrial DNA as my brothers and my sister and my mom. My kids don't have my mitochondrial DNA. And what I usually like to tell people as they tour through the FBI, men are mitochondrial DNA dead ends. We're completely worthless. Depending on the audience, you get chuckles, or sometimes you get a blank stare, but it's the God's honest truth. There have been rare occasions of observing some degree of paternal mitochondrial DNA, but for the most part, mitochondrial DNA will be inherited along maternal lines. Mito's in a small circular structure, and unlike nuclear DNA, you typically can find hundreds to thousands of copies per cell. That depends on the cell type, and that's a tremendous advantage of mitochondrial DNA. It allows us to look at different sample types, hairs, teeth, bones, really compromised tissues or remains, or even compromised blood samples or saliva samples that have been stored incorrectly. With mitochondrial DNA, we're not looking for any series of repeats or length variants. We're looking for the sequence of bases. So we all know there are 4 bases, G, C, A, and T. The order of those 4 bases for me may be different from the order of Tim's. I remember you from our Mido school. So I'm not picking on you, or different between Laura and I. That's what we're looking for. The fact that I'm different from someone else is a very powerful and informative piece of information. If Laura and I have the same type, then different questions need to be asked, and there needs to be a different treatment of that information. This is the tried and true gold standard that Steve had referred to before, sequencing. Getting to this point is really a tremendous task. Everybody knows the procedure and how much time and how much care it takes. But in the end, you can have a beautiful sequence of bases and be able to independently and discreetly identify the base location and provide what we call the mitochondrial DNA type, or the mitochondrial DNA sequence. After deriving the sequence, the order of the bases, these complementary strands are aligned to the revised Cambridge reference sequence. Differences to that reference sequence are then described. So essentially, we're taking what I like to say, and the analogy I like to give, is that we have a set of driving directions, maybe going from Washington, D.C. up to New York City to visit my in-laws for the 10th time in a month. So as I'm driving north, I start out in Manassas, Virginia, and I eventually get on I-95, and I pass through New Jersey, and I get all the way up to New York, and I know the exit that I'm going to get off. I don't pay attention to the exits in between. I just pay attention to the road maps and the markers that I need to know about in order to make my next move. It's essentially the same. We're looking for differences compared to a standard. Mitochondrial DNA has been fully characterized, approximately 16,000 bases, give or take some insertions and deletions. There are generally 2 regions, the control region and the coding region. As you can imagine, the control region is the site of replication and transcription. There's a high degree of variation clustered, packed within this control region. Specifically within 3 regions, hypervariable region 1, 2, and 3. With the most information, what I mean by information is the most variation between individuals occurring and being packed within HV1 and HV2. But keep in mind that the coding region takes up the bulk of the genomic space. The control region itself is about 1.1 kb. The rest of the genetic information is out in the coding region. This is where you have your oxidative phosphorylation proteins, ribosomal RNAs, transfer RNAs, and you can have a tremendous amount of variation within these regions. The key is that this variation will be scattered across that entire expanse of real estate. So having technologies available to maximize that variation is very challenging. As I said before, tremendously high copy number. Interesting about mitochondrial DNA, produces a polycistronic message. So after transcription, it produces a huge RNA that then gets processed after translation, after transcription. It utilizes a different genetic code compared to nuclear DNA, and it also possesses a relatively high mutation rate. And this is due to the use of polymerase gamma, which in itself has a compromised repair mechanism. This leads to that tremendous amount of variation between you and I. Again, it's inherited through your mom, and it's only a single locus. It's only a single genetic element. Bases are not independent, meaning that polymorphisms that get described are not independent of each other. Whereas with nuclear DNA analysis, you can take your results from location 1, multiply that frequency by location 2, 3, and 4 across your different chromosomes. You have to treat mitochondrial DNA as a single locus and just treat it as a bulk piece of information. And that has an impact on our statistics. Mido was first discovered in the early 1960s. Back in 1959, there was some research being done on what's called Luff's disease. It's a mitochondria deficiency within muscle tissue. The sequence for mitochondrial DNA was published in 1981, described as the Cambridge reference sequence, and as you can guess, it came out of Cambridge. Sanger was one of the authors of that paper, Sanger from Sanger Sequencing. The sequence itself was revised in 1991. There were some discrepancies found within the 1981 description due to the way the sequence was generated, different lab techniques. So what we use now in our analyses is the revised Cambridge reference sequence, RCRS. And Mido is used in several different disciplines. Medical scientists use mitochondrial DNA to study the inheritance of diseases. Diseases such as Leber's hereditary optic neuropathy. There are all different types of muscle myopathies associated with it. Diseases that typically are impacted in high energy demand tissues. The more mitochondria you have, the more energy gets produced. So tissues that require a lot of energy get impacted greater than other tissue types. Evolutionary biologists can use Mido to compare us to different species, to where we evolved from. And anthropologists have used Mido to look at the migration of people. How have people migrated throughout time and throughout geography? Mido's been used in the forensic community since probably the early to mid-90s. The FBI lab was not the first place to do a mitochondrial DNA exam, but we were one of the first places to initiate the research and the feasibility back in 1992. Our first court testimony, and actually the first admission of mitochondrial DNA, was in August of 1996. For us, this was a foundation event. So here we have mitochondrial DNA being introduced and challenged in court. It was used in the case of Tennessee versus Paul William Ware. Paul William Ware was accused of sexually assaulting, raping, and killing a young girl. What was analyzed was a hair recovered from this girl's throat. The mitochondrial DNA type from that hair was in concordance or similar or the same as Paul William Ware. And you'll notice that there are different terminology that needs to be used with mitochondrial DNA. It's been used in international cases. I myself have testified in Canada. That's my most exotic trip so far. And it's been accepted federal, state, local jurisdictions, and there have been several appellate decisions on all different levels. So the foundation of Mido and the application of it has been widely accepted in court purposes. It's been used with several different types of identifications. Now keep in mind, mitochondrial DNA is not unique. Mitochondrial DNA by itself in a vacuum cannot responsibly be used to identify an individual. So with any of these situations described here, with the Vietnam unknown soldier, Mido was used in conjunction with other information, whether that information is anthropological, whether it is a closed system, so an airplane goes down and you know that the passenger list contains 5 people, and you compare those 5 people's relatives to the sets of remains. This is all non-DNA based information that can lead to an identification. The case of Tsar Nicholas II and the Romanov family was a fantastic example of the use of mitochondrial DNA across several different generations. Anna Anderson Manahan put herself out as the lost Tsarina. related to this Romanov family, and it turns out her mitochondrial DNA type was different from the rest of the Romanov family. Jesse James is another interesting example of a combination of nuclear and mitochondrial DNA used together to identify a set of remains. And the September 11 attacks. Mido was used in conjunction with a lot of other different technologies and circumstantial information and evidence to help identify the victims of that event. Mido is fantastic. I'm a big fan of it. Why? You can get down to approximately 2 millimeters of hair and get full mitochondrial DNA information. And what I mean by full mitochondrial DNA information for our purposes is that we can amplify regions of HV1 and HV2, approximately 600 bases in total, and get full sequence information for 600 bases from 2 millimeters of hair. We can work with pretty degraded bones and teeth, items that have been subjected to incredible conditions, and I'm sure John Plans can describe some of that work, dealing with the types of cases that he's dealt with. Degraded samples, so again, tissues. More and more frequently, we are being confronted with types of human remains that have been involved in explosions, in tremendous fires, high pressure, high temperature. Is the sample a mixture? Now, this is one of the key considerations with mitochondrial DNA as it currently stands. Sequencing of mitochondrial DNA is really challenged by a mixed mitochondrial DNA type, mixtures from two different individuals. And I'll go into more of that in my mixture talk, but essentially, mixtures are very challenging to interpret. So they may not be the most suitable item to subject mitochondrial DNA analysis on. What are your available reference types? If I decide to go and rob the counter out here and lift every ice cream bar that's out there, and I wear a ski mask and I'm charging out the door and I leave it behind, or it gets torn off because Steve's chasing me because he left his credit card there for me, and I hit the road and I wind up in Tijuana, well, you know, if they get that mask and they recover some hairs from it, those hairs can be compared to any of my brothers, my sister, my mom, because we all are expected to have the same mitochondrial DNA type. So it's valuable in criminal situations such as that. It's also very valuable when it comes to unidentified remains. If you don't have a direct reference sample, say I don't leave a toothbrush behind and all of a sudden I disappear and you find a set of remains that fits my description and all the circumstances surround it are pretty equal, you can go to any of my relatives again and get a suitable reference type. The variation that you find between people will depend on the background of the individual, the history, the ancestry of that individual. Bruce has found through his publications that Caucasians differ by approximately 8 bases. There's always the possibility that you and I differ at a single location, but in general, we expect that people that are unrelated to differ by 8 bases at least. And that we can take advantage of with the mixture interpretation. From the very beginning, it must be understood, Mido is less discriminating. You can possibly achieve, unique identity through statistics with nuclear DNA. Currently, our best or most discriminating statistic is 1 out of approximately 600 individuals may be the source of this mitochondrial DNA type. Cannot currently interpret mixtures, at least with the technology that we're using now. With sequencing technology, mixtures are uninterpretable. And there are some key things to keep in mind other than the fact that it's less discriminating and mixtures are challenging. These things are important for Mido especially, things like contamination, heteroplasmy, and the statistics, the databases that we use to supply some type of weight to the comparisons that we do. Now, things like contamination and statistics, they're valuable and extremely important with nuclear DNA exams. Heteroplasmy is unique to Mido. But with the technologies that we're doing and the techniques, the contamination is really an important key. Typically, we follow 8 steps, or general labs follow 8 steps. You go through an extraction process, you go along with your amplification. The amplification conditions are such that typically high cycle number, Anywhere between 30, 32, 36, maybe 40 cycles are used. So this is a key part in dealing with contamination. For us, we do a post-amplification quantitation. The key part of this quantitation, not only to figure out how much mitochondrial DNA did we synthesize through our amplification, but also to evaluate some of our controls. These controls are going to tell us, have we introduced any DNA into this process? Have we contaminated our situation? And then we go through the technical parts of performing a cycle sequencing reaction, doing a controlled synthesis of DNA. And by controlled, I mean that we're introducing di-terminator nucleotides with fluorescent tags attached to them so that we can identify, after this stretch of DNA, what was incorporated, an A, a G, a C, or a T. Those fragments are then separated currently by capillary electrophoresis, and then we go through the process of looking at that sequence, comparing it to our reference, our revised Cambridge reference sequence, and then comparing sequences against each other and determining what answers, can we answer any questions at all? Can we say, is this individual a possible source of this item, or are they excluded? And based on those interpretations, we may need to go and assign weight. This is a typical example of how we display these results. We describe the range of nucleotides that are examined and the different polymorphisms relative to the revised Cambridge reference sequence contained within that sample. And if you take a look, we examine and compare a question item against our known items. And the question that we're trying to answer, can I exclude any of these known items, any of these individuals, as being the source of this question item? Now, how do we go about doing that? It's actually very simple. You look at your known item, and you look at the polymorphisms that are present, and you do a comparison. You say, are the polymorphisms contained within known item 2 the same or different as the polymorphisms contained within the question item? And if you look at the question item compared to known item 2, you can see that these are different. It's a very simple interpretation. I can exclude the source of the known item 2 as being a possible source of the question item. And if you look at known item 1 and compare that to the question item, we'll see that there's a common base at each position. At 16,111, there's a T in the question item, 16,111T in the known item, and so on with all the rest of these polymorphisms. So the interpretation would be, I cannot exclude this individual as being a source of this question item. Now, notice that the language is very important. It's cannot exclude. It could never be, this is the source. And we have a third category, inconclusive. This factors into the phenomena of heteroplasmy and also the interpretation guidelines that are in place. For an interpretation of exclusion, we need to find at least two base differences between a question item and a known item. Two differences because of heteroplasmy, and I'm going to discuss that now. Well, next slide, or the one after. If we have a situation of cannot exclude, so the evidence and myself have the same mitochondrial DNA type, what does that mean? Do I have a type that's very common in the population? Do I have a type that's very rare in the population? Well, we look in a population database. We have a collection of mitochondrial DNA types. Some of these mitochondrial DNA types are pretty common, like these green people over here. Some of them are pretty rare, like this hatched one. Well, here's me, here's my type, and what happens is someone takes a tally and figures out how many times has Les's type been seen. Has it been seen several times? Has it been seen once? Has it been seen at all? Now, if it's been seen several times, easy. We just plug that information into a simple statistical calculation. What we need to do is make sure that the results of our database are reflective of the general population. And for us, it's the general US population. So we do that by applying a 95% confidence interval around those number of observations. Keep in mind that within the database that we currently have, most of the sequences occur a single time. Okay? The true population frequencies of the majority of sequences is unknown. And we'll never be able to truly know that unless we go out and sample everybody. And it's just not going to happen. Frequency estimates are based on database size. whether you do a confidence interval dependent on 5 observations or 0 observations, it's still going to be based on the number of people within that database. So as the databases get larger, you expect to have a more reflective collection of people in the general population, and your estimates will be closer to the actual value that you would find. And here's our friend heteroplasmy. Very simply, heteroplasmy is the presence of more than one mitochondrial DNA type in a person. It is operationally defined, meaning that depending on the technology that you're using, you will either be able to detect and observe heteroplasmy or it will not be detected by you. For sequencing, well, with Mido, there are 2 types of heteroplasmy. There's sequence heteroplasmy. It's referred to as sequence heteroplasmy. Point heteroplasmy can be referred to as a SNP, a single nucleotide polymorphism. This is the presence of more than one base at an individual location. So in this example, you can see that there's a presence of red and blue at this particular location. It's a mixture of T and C. And what's important in the interpretation is that what you're observing in the forward strand, which is below, is the same as what's in the reverse strand. It's important to have 2 different strands and to take the entire sequence into context. What you observe in the forward strand needs to be reflected in the reverse strand. There is known to be some kind of sequence content impact on your ability to incorporate diterminators. So if you see some type of blip in one of your reactions and not in the other, most likely it's due to something going on in the chemical reaction of your reverse strand that's not reflected in your forward strand. Now with length heteroplasmy, in mitochondrial DNA, it can be found at a couple different locations, most commonly in HV1 and in HV2. And this is an example of length heteroplasmy occurring in HV1. There's your forward strand, reaches this point, and then you have this overlay of sequences that really becomes very difficult, if not impossible, to interpret. And here's a reverse strand. You can see that you reach a certain point, and the data just essentially falls apart. Okay? So in terms of information content, we can assign a value or an identity to this base. We can describe it as a mixture of C and T. In terms of information within this stretch, we can go up to a certain point and then essentially say, don't know what's out there. Why? Because you can't really interpret that information, that mess. If you have items of evidence that you're looking to compare against each other, what do you do if you have a situation like this where you find you could have a mixture in a C and a T there? Well, our practice at the FBI and throughout most of the Mido community is that you treat this location as a C and a T, as a combination of the 2. So if you do a comparison and your known type has a T, well, you say there's a common base there. What's common? The presence of a T. Because heteroplasmy is operationally defined and it also has some tissue-specific expression, you need to treat this almost as a wild card and say during your comparisons, if I have the presence of one of these bases in my known type and everything else is the same, then these 2 are concordant, common base, and I can't exclude the known type as being a possible donor of this question item. Now with length heteroplasmy, it's a little bit different. Because we're not gaining any extra information in HV1, we just count up the number of Cs and type it according to the Cambridge reference sequence. HV2 has a different structure where there's a stabilizing T in the center of these Cs, this stretch that leads to the overlapping sequence. So with length heteroplasmy, you're going to look for commonality in the number of length variants. Do you have species that demonstrate the same number of C residues or the same structure? Do you have overlapping length variants between your question item and your known item? Now, contamination is a huge challenge and something that every Mido lab needs to keep in mind from the beginning towards the end. And probably in our own lab, we spend 75 to 80% of our work trying to minimize and monitor for contamination. So everybody gowns up in personal protective equipment, so face masks. Depending on the sample type, you may wear a head bonnet, full lab coat, constantly changing gloves. Tremendous cleaning procedures. Most of you in the room can describe this at length. How many times do you clean out your hood with bleach? How many times do you have to wipe off your pipetters? How long do you have to throw something in the stratolinker to expose it to UV light? dedicated equipment. We don't share equipment between post and pre-amplification. We actually, and some labs actually have dedicated rooms separating your question items from your known items to make sure there's no chance of carryover. And that's important because you don't want any residual product from your question item to ever be challenged and be accused of ever cross-contaminating something. And monitoring is a huge step in the process, using all of your controls, so reagent blanks, making sure that the chemicals that you use in your extraction have been checked to see if any DNA is present within them. Negative controls initiated at amplification and carried throughout the entire process. And then, because of the number of cycles and the copy number of mitochondrial DNA, you have to think about, well, what do I do if I do detect contamination? Does that mean that I can't use the mitochondrial DNA type that I developed from this evidence? There has to be some kind of criteria determined. Can I interpret the contaminant? Can I exclude the contaminant as being the same as the evidence? You'll see a little bit of variation from lab to lab in what they've decided to go with, what process, what procedures, how they're going to handle contamination. As I said before, compromised samples, cold cases, are fantastic candidates for mitochondrial DNA. We regularly get calls about 30-year-old cases. Hey, we have some hairs that have been recovered, that have been preserved on slides. We developed a subject. He's actually dead, but his brother's around and he's willing to give a swab. Do you think you can do a comparison? Having available reference items, whether you're doing a missing persons type case, trying to identify unidentified remains, or if you're just trying to associate someone to an event or a crime or a situation. And more, outside of missing persons cases where we look at kinship, we get confronted with questions such as, could person A and person B be relatives of each other? Could they possibly be cousins, siblings, or something else? Because we have this information telling us that they're brothers. And we would like to leverage that when we go and take a talk with them. So Mido, in combination with other technologies, WISE and other circumstantial evidence, can really be maximized. Now, I have one slide for future. I know Bruce is going to have more. And I hope I don't steal Bruce's thunder. Right. I see an increased demand in Mido. Why? Because word is getting out. More and more laboratories are doing mitochondrial DNA. Cold cases are still cold, and they're still being dug up. I see population databases increasing in size, becoming more reflective of the general U.S. population, and also moving out into international communities, specifically allowing us to handle different types of events internationally. Some places are already looking at the variation found within the coding region. Some of the work has focused on trying to elucidate out the most common mitochondrial DNA types, focusing on the types that you would probably encounter more frequently. So for example, the most common Caucasian type is found approximately 8–9%. So with the goal of trying to break that down a little bit, some labs have decided to look at variation outside of HV1 and HV2. More and more automation will take place, not to push people out of jobs, but just to make sure that you remove a human interaction, you remove that possibility of an inadvertent contamination, and you free people up to do the more time-consuming and technically challenging procedures, such as handling a hair and transferring it from tube to tube and going through that extraction process. And there will definitely be more technological advances. You all know that we're here to discuss mass spec. I'm a big proponent of this technology. Lois had said it early in the day that this is very exciting. But there are other technological advances that are occurring right now. There are chip-based hybridization techniques. There are microfabricated wafers that can do capillary electrophoresis on a 4-inch wafer. So there are other advances that are going to be out there. And in total, I think that's going to have a huge impact in our ability to generate this type of data and provide some type of information. If there's ever any question, I'm sure you're all so well versed that there aren't too many questions. But please feel free to drop me a line. No question is too simple or too difficult. If I can't answer it, as I usually can't, I'm surrounded by a lot of smart people. So please feel free to reach out. Thanks.